All right, ladies and gentlemen, welcome to your cell transport notes. By the way, one of my big pet peeves is people talk about learning or doing something through osmosis. Yeah, that's not how that works. That's actually just diffusion of water. So don't make that dude look at you with that facial expression. All right, so to talk about transport in cells, first we need to get a couple of terms down. Hopefully you learned these in chemistry, and if not, then let's talk about them now. So there's a difference between a solute and a solvent. These are all dissolved, they're in a solution, but the solutes are usually some sort of particle, molecule, ion that is dissolved into a solvent. So in this case, you have these little particles, these little orange particles, those are your solute, and the liquid that they are dissolved in is the solvent. In our case, since we're looking at biological systems, the vast majority of the time our solvent is going to be water. So we can use the terms water and solvent interchangeably because they pretty much mean the same thing here in bio. So we're going to talk about the different types of transport, and I hope this starts to come back to you from your freshman year. But passive transport is the type of transport that does not require any energy put in on the part of the cell. So this can happen naturally, and it happens without any extra energy being put in. This is the net movement of molecules from a high concentration to a low concentration. So notice what we have here is we have this hexagonal shaped molecule that seems to be able to cross the lipid bilayer, and notice that it's moving from where there's a lot of it to where there's less of it. Basically, I think of it that these particles move until they spread out. Notice that our final configuration has these particles evenly spread out on both sides of this membrane. When they are evenly spread out, that is called equilibrium, because we have equal concentrations on each side. Now, while you do have equal concentrations on each side, that doesn't mean that these molecules have stopped moving. That just means that pretty much for every molecule that moves from one side of the membrane to another one's going to cross in the opposite direction. So molecules can still move, molecules can still cross, but they're not changing the overall concentrations. So there are a couple of different types of passive transport. There is simple diffusion, and this is applying to things that can move easily through the cell membrane. Then there is facilitated diffusion, which this is for stuff that cannot move easily through the cell membrane. Usually the membrane is impermeable to these. That would be ions, that would be glucose, and that would be water. These guys require proteins in the membrane to get them across the lipid bilayer. So here we have an example of facilitated diffusion. This is a glucose transporter. So it is a molecule that binds glucose. So notice it has a binding site that's shaped like this glucose molecule. It's got a high affinity for glucose. When glucose comes in and binds, that causes this configuration change in the protein, which opens it up to the inside of the cell, and that configuration change also decreases the binding affinity for glucose, so that's why the glucose pops off into the cell. When the glucose is gone, the configuration of the protein goes back to the original shape, so that way if it encounters another glucose molecule, it can go ahead and take that in. None of this required energy on the cell's part, which is a good thing because you're taking in glucose to produce energy, and it'd be kind of pointless if you did that and you had to spend a lot of energy to get it into the cell. There is a very special case of diffusion. It is osmosis. This is the diffusion of water, and this will happen usually when you have a solute that cannot cross over through a barrier, so water instead will cross. So the solvent is crossing instead of the solute. So notice in this diagram here, we have these large sugar molecules that are too big to cross through these pores in the membrane. So instead, water molecules will cross. Now notice we have a low solute concentration, and I think of it as a relatively high solvent concentration on one side. Then we have a higher solute concentration, and then a lower relative solvent concentration on this side. Notice then that enough water molecules move over towards the right that the concentrations are kind of even. So like I said, things like to spread out until they're balanced. So this leads us into a moderately complicated topic of water potential. So water potential involves knowing about hypotonic, hypertonic, and isotonic solutions. So this should be familiar from your freshman year of biology, though it may have been confusing back then. And it has to do with what happens to cells that are placed in different solutions. Think about this. I think of tonicity as the amount of stuff, amount of solute. So you can think of it as a stand-in for concentration. What does hypo mean? Hypo means under. What does hyper mean? Hyper means above. So what this means is hypotonic solutions have a lower solute concentration. Hypertonic solutions have a higher solute concentration. And this is important because remember, water moves from where there's a lot of water and less solute to where there's more solute and less water. If we have a lower concentration of our solute outside of the cell, that means we have more water relative to the inside. So that means that water is going to move into the cell. And that's when we talk about how animal cells in these kinds of environments will pop or lyse. On the other hand, in a hypertonic solution, there is a higher solute concentration, a lower solvent concentration, and therefore water is going to move until it tries to even things out. So it's trying to make the outer environment less concentrated. So water is going to leave the cell. And that is not a good idea for any type of cell. Animal cells do best in an isotonic solution, iso meaning equal. So when the solution outside of our cells has the same concentration of solute and stuff as the solution inside of our cells are in their ideal condition. So we're going to define water potential now. Water potential is literally the ability of water to do work. It's its potential energy. So it's basically the free energy per every mole of water. So what this depends on is how free are the water molecules? Can they move around? Can they do work? And what would keep a water molecule from being free? Well, if we look at this picture, this is a salt crystal that's been stuck in water. So these are alternating positive and negative ions, and these are water molecules. Over time, what's going to happen is these ions are going to end up mixing with the water. So notice that these negative oxygens are clustered around this purple, so it's got a positive charge, and the positive hydrogens are clustered around the negative green ion. So notice these water molecules are now tied up. They're not free to do any work. Neither are these guys, neither are these guys. So the more solute that you add in, the more solute that's dissolved, the less free water you have. So this is water tied up, this is water tied up, tied up, tied up. And so as you add more solute, the water becomes more and more tied up. There's less free water, and therefore you're decreasing the potential energy that the water has, and you are decreasing the water potential. So how do we measure water potential? The units aren't quite as important because you're not going to have to really put units on the AP test unless it's a free response question, and you don't tend to see those with water potential very much. But you can measure it in megapascals and a unit of pressure, basically. This symbol right here is called psi. It's a Greek letter, and we use it to represent water potential. So whenever you see that letter that looks kind of like a trident, you're looking at water potential. Now, if you have pure water in an open container, it's got a water potential of 0. Once you start adding solute to that solution, you're going to have a more and more negative water potential. It's going to start going down because those solute molecules tie up that free water. Okay. So, like I said, tonicity is basically going to affect osmosis. Hypotonic solutions have a higher water potential. Hypertonic solutions have a low water potential. Remember, when you add solute, that's going to cause your water potential to become more negative. So if you are hypertonic, you have a lot of solute in you, so you're going to be like a really high negative number. You might be like -5 or -11. On the other hand, if you have a hypotonic solution, it's going to have less solute in it, so that might be, so this is hypertonic, and a hypotonic solution may have a solute potential of like -2. So -2 is larger than -5. Remember, with negatives, the farther you get from 0, the smaller the number you are. And water always moves from hypotonic solutions to hypertonic solutions. So it goes from a high water potential to a low water potential. So this is the direction that water would move. So it would go from the hypotonic solution to the hypertonic solution. So this is just showing you animal cells and what happens in each of those solutions again. If we relate it back to our tonicity, these tonic terms are always comparative. So we have a solution that's hypertonic compared to another solution, because hyper and hypo mean like more and less. So here we have animal cells that are in a hypertonic solution, which means their interior. So let's say this is the cell. The interior is going to be hypotonic. The exterior is hypertonic. Water always moves from hypo to hyper. That means it's going to be moving out of the cell. The cell shrivels and dies. If cells are in a hypotonic solution, that means the outside is hypotonic. That means the inside is going to be relatively hypertonic. Water always moves from hypo to hyper. So more and more water keeps going into the cell. Eventually it's too much for the cell membrane and the cell pops. This is different in plants because plants have that nice rigid cell wall surrounding them. So they act the same way in hypertonic conditions because the inside of the cell is more relatively hypo. In isotonic conditions, they have as much water entering as leaving, but that's actually not healthy for a plant either. Their most ideal condition is to be in a hypotonic solution because their cell wall allows them to take a lot more pressure of water trying to go in, since it's more rigid than a cell membrane. So when they're in a hypotonic solution, more and more water keeps trying to rush into the cell. It fills up this central vacuole, which helps push out against the cell walls and helps keep this cell maintaining its shape and helping the plant overall maintain its shape. So how do we calculate water potential? You do have this equation on your formula chart. So that's water potential equals pressure potential plus solute potential. And we're going to talk about each of those. The solute potential, which is also called the osmotic potential or Psi-S, measures the effect of the solute that's in there. Makes sense with the name. So as you add solute, the water potential drops and the water will tend to move into that solution more. We calculate it using this formula, which is also on your formula chart. Your formula chart also has the meanings of each of these terms. So R is a constant. It will always be this number. T is your temperature in Kelvin. So make sure that they'll tend to give you a temperature in Celsius. You're going to have to change it over to Kelvin, but you just do that by adding 273. C is your molar concentration. They usually either give that to you or it's what you're solving for. And I is your ionization constant. So if you think about it, Na and Cl. Sodium chloride dissociates or breaks up into a sodium ion and a chloride ion when it goes into solution. It's got an ionization constant of 2 because it forms 2 distinct entities, each of which is going to tie up some water molecules. On the other hand, if you had H2 PO4, and don't quote me on whether or not that's an actual proper chemical formula, but it's going to dissociate into 3 different ions, a couple of H's, and then I don't know if that charge is right, but whatever. So it's going to dissociate into more than one particle, so it's got an ionization constant of 3. Whereas NaCl has an ionization constant of 2. Something like glucose that's not ionic has an ionization constant of 1. Notice that there is a negative sign in front. It's a little hard to see on your printed notes. So it's negative ICRT. So these are the practice problems that I'm asking you guys to complete in your notes. We're going to go ahead and complete them now. So we need to know the solute potential. And we're given some information. We know it's sugar. Sugar does not break apart in water, so it's got an ionization constant of 1. We're given the molar concentration, and we are given a temperature. Now notice our temperature is in Celsius. We need to convert it to Kelvin. So that means we need to take our 27, add 273 to it, and that's going to give us 300 degrees Kelvin. So our solute potential is negative I times CRT. It's going to be negative 1 times 0.3 times 0.0831 times 300. If you go ahead and multiply that out, and on the test, remember you do have a calculator in part for this, that's going to equal negative 7.48. And that is your answer. That is your solute potential. All right, let's go on to number 2. So number 2 asks you to calculate the water potential of a solution of 0.15 molar sucrose in a beaker at 20 degrees Celsius. So it helps if we know our equations. So I'm going to just go ahead and write them out here. So our total water potential is going to be our pressure potential plus our solute potential. Our pressure potential, if we're in an open beaker, which it looks like we are, is going to be 0. Therefore, our water potential is going to be equal to our solute potential. Now, what is our solute potential? We're not given that, but we are given a molar concentration and a temperature, so we can calculate that. So our solute potential is going to be negative ICRT. Sucrose is a sugar, so will it ionize in water? It should not. So I should equal 1. C is our molar concentration, is 0.15 molar. R is that constant, it's always 0.0831. And T is our temperature in Kelvin. So if we're given our temperature in Celsius, we need to add 273 degrees to it. So that's going to mean that our temperature is 293 degrees Kelvin. We need to take all of that and we need to multiply it out. So that's going to be negative 1 times 0.15 molar times 0.0831 times 293. And when I multiply all of that out, then I get negative 3.65 as my solute potential. And remember, I said my total water potential equals my solute potential. So the water potential of this solution is negative 3.65. All right, let's do the third question. You measured the total water potential of a cell and find it to be negative 0.24 megapascals. Or millipascals. If the pressure potential of the same cell is 0.46, what is the solute potential of that cell? So for this, we're probably going to use this equation. Our total water potential equals our pressure potential plus our solute potential. We are given our pressure potential. We are looking for our solute potential. So our total water potential is negative 0.24. Our pressure potential is 0.46. So we need to solve for our solute potential. So when I do that, what I can do is rearrange this equation. So that way I have the solute potential on one side of the equation. So it's going to be equal to my total potential minus my pressure potential. So it's going to be negative 0.24 minus 0.46, which is going to be negative 0.7. And that's our pressure, solute potential, excuse me. Number 4, a student calculates the water potential of the solution inside a dialysis bag at 22 degrees Celsius to be negative 6.25 bars. Bars is another unit of measuring pressure, and the solution surrounding the bag to be negative 3.25 bars. So, what is the direction of net water flow? So we have a beaker or a cup or whatever. It has fluid in it. The fluid has a water potential of negative 3.25. You have dialysis tubing inside. And that dialysis tubing has a potential of negative 6.25. So water always moves from where there is a greater water potential to where there's a lower water potential. So where is my lower water potential? My lower water potential is inside of my bag, because the farther away I get from 0, when I'm at negative numbers, the smaller the number. So water is going to move from where I have that higher potential, which is outside of the dialysis bag, to the inside of the dialysis bag. Okay? Number 5, if a plant's pressure potential is 2 bars and its solute potential is negative 3.5 bars, what is the resulting potential? For that, we're going to use this equation again. And so I've got values for my pressure potential. It's 2 bars. And then I've got values for my solute potential. It's negative 3.5 bars. When I add those together, my total potential is going to be negative 1.5 bars. Okay, the math on this is fairly uncomplicated. What you really need to be able to wrap your head around is the direction of water flow and also being able to visualize what these numbers mean. So let's talk about pressure potential. In a plant, it's also known as turgor pressure. Plants are very highly reliant on manipulating their water potential, because that is what actually drives the movement of water up a plant and from the roots up to the tips. So this pressure potential in a plant, a lot of that is supplied by the force of the cell wall pushing inward. So here you'll see a plant cell. When it's put into distilled water, notice it's got no pressure potential because there's no pressure pushing out on the cell wall and there's no pressure pushing back in from the cell wall. However, when you put it in distilled water after a while, enough water rushes in that the pressure potential is going to increase. Okay? So, turgor pressure is the pressure of the cell insides pushing against the cell membrane and the cell wall, and that is equal to and opposite in direction to the wall pressure. The wall pressure is going to basically be pushing back against the turgor pressure and canceling it out. If it didn't, that cell would either pop open or it would shrink. And eventually, after a cell has been in a solution for a long time, the water will move until they've reached an equilibrium, and at that point, they are going to have an equal water potential. So notice that we have this potato cell in pure water, which has a water potential of 0. It's got no solute, and it's in an open container. Our potato cell initially has a negative solute potential because it's got stuff inside of it, so its potential is negative 3. Eventually, what's going to happen is enough water is going to move into the cell to expand its pressure potential, so that way its total potential is going to be the exact same as the water's. So everything is going to be in equilibrium after a while. So what I was talking about is the fact that these differences in potential are what drive water to go through a plant. You probably, or you may have heard in biology class your freshman year, that it was transpiration that drives water up a tree or up a plant, but it's actually this difference in water potential. So plants can manipulate their water potential in their cells by either adding or removing solute. So they change their solute potential, thereby changing the potential of cells in the roots, cells in the stems, cells in the leaves, and thereby they're able to kind of create this decreasing set of water potentials as you go up the plant, until finally when you get to the atmosphere, that's the lowest water potential of all. Remember that water moves from a high water potential to a low water potential. So the highest water potential on here is in the soil and the lowest is in the atmosphere, so you're going to have a net movement of water in this direction. So it's going to go from the soil through the plant and into the atmosphere. This is visualized right here, so you can see the water potential of all of these things. The water potential of the soil is negative 0.3 MPa. Notice that it gets lower when you go into the xylem in the trunk. As you go higher, it gets lower. Inside the leaf, it's even lower. Outside of the leaf cells, but still within the leaf, it's lower, and it's much lower in the atmosphere. So water is constantly going to be moving from the soil in, up the plant to the leaves, and then out to the atmosphere. If you have soil that is too dry, that has no water in it, you don't have this water potential difference, then you stop the flow of water into the tree. Trees don't have hearts, they don't have anything to pump water around, and they rely on this to be able to have a constant flow of water and the nutrients and things that are dissolved in it to keep themselves alive. So you may have forgotten that we're actually still talking about diffusion when we took that sidetrack onto water potential, but we are still talking about diffusion. So the last type of diffusion is facilitated. You can use either just a channel, which acts as like a hole in the membrane that allows certain molecules to pass through. These channels are usually highly specific for a certain molecule. So for example, we looked at aquaporin in that membrane model. Aquaporin has like electric and chemical properties that make it attractive to water and water alone. Water can pass through, other things like ions cannot. And so it is very specific for water. So it's not like just opening a hole in the membrane and letting everything go in and out. It's opening a very specific doorway. And then you have carrier proteins. The difference is they don't have this full-on opening constantly through the membrane. Instead, they shuttle molecules across the membrane, usually by changing conformation or shape. All right, so now we're going to talk about the other type of transport. So this isn't passive transport, instead it's active transport, and I'm hopeful that you remember the difference between the two. So active transport does require energy, and this energy in a cell is going to be in the form of ATP. Because it's using energy, it's moving molecules against the direction of diffusion. We also say it's against the concentration gradient. And that's moving from a low to a high concentration. There are quite a few techniques of active transport that do use membrane proteins. They're very important for active transport. So the simplest form of active transport is usually you use some sort of pump to actively move ions, molecules, etc., to a certain side of the cell. So there are two major pumps that we will need to talk about this year, one of which is the sodium-potassium pump. This is especially important in neurons. This is what creates a concentration gradient of ions, and it's what creates charges that can then move into and out of a cell during an action potential. So what it does is it basically spits out sodium and brings in potassium. Then what's going to become very important for photosynthesis and cell respiration is a proton pump. A proton pump is going to pump these protons, which are just hydrogen ions, out of the cell or out of a compartment, and they're creating a concentration gradient. Then that concentration gradient can then be used to generate some energy and create ATP. So we're going to look at proton pumps and proton concentrations quite a bit later in the year. These next two major forms of active transport are termed bulk transport because they're going to move large amounts of things. And these are going to involve folding and unfolding of the cell membrane. So endocytosis is literally taking stuff into the cell. It's got two forms, well, three major forms. Phagocytosis literally means cell eating. And so that's taking in solid particles. If you have a single-celled organism, this is how they ingest particles or other organisms. They will basically form what's called an invagination of the membrane, where it basically kind of pockets itself in. Then that pocket kind of closes off and forms a vesicle, which then can go into the cell and be digested, etc. Pinocytosis, on the other hand, literally means cell drinking. And this is when a cell takes in a sample of the extracellular fluid. For one reason or another, there may be a high concentration of some sort of molecule out there that it's trying to get into the cell, but it's going to perform a similar procedure where you have this invagination or infolding of the membrane that pinches off and then forms a vesicle that can then move throughout the cell. There is a third type of endocytosis called receptor-mediated endocytosis. It involves this chemical called clathrin. You don't need to really know much else about clathrin other than the fact that it has some signal molecule bind, and which signal molecule it is depends on the type of cell and what it's trying to get into the cell. When that signal molecule binds, that triggers this infolding or invagination of the cell membrane, which then kind of buds off and forms a vesicle. So the similar thing between all of these forms of endocytosis is, of course, they're all taking things into the cell and they're all forming vesicles. And exocytosis is just the opposite process, where you have a vesicle that's going to basically melt into the cell membrane and release its contents out into the cell. And that's just a point we're going to talk about more when we get to the cell organelles, which will be our next lesson. So part of the reason why eukaryotic cells are able to become much more complex than prokaryotic cells is because we have all these internal membranes that do a lot of jobs inside of us. I hope that's helpful. Go through those practice problems. Make sure you know how to do them, because we will be doing something with water potential in class. Thank you. Good night.